So this has been going, and with Marcy's leadership, I mean, this has been going for a long time, and it's really holding steady. You know, you'll have to give us some feedback on whether we're doing, you know, I just got off the phone on a cardiomyopathy, cardiac disease ontology call from the European Bioinformatics Institute. And, you know, I'm looking at this pizza over here, and maybe I ought to call back and say, hey, where did you put the nutrition term? But so let us know about the food. We can change it. Maybe we can even get more healthy. You know, I had a chance to, Ari he gave a nice bistro recently. You know, his thesis is starting to come along nicely. He's been working very hard with Jerry and I on getting this paper out the door. And I had a chance just to, you know, look at his PowerPoints, I don't know, 10 minutes ago. And, you know, I suggested, if he corrected me on one thing, but I got him to take out something I thought was bad.
Good.
You know.
I know that was too.
Overall, you know, what I saw is a two-part talk. One is giving some, you know, kind of informed opinions on GWAS and where that's going. And GWAS has really been changing over the years. The emergence of EQTLs and MEQTLs and all this is a big deal. And, you know, what we were able to get out of GWAS, you know, 10 years ago versus now is really quite different. And that's, you know, and with Jerry's leadership, we've been able to, you know, push the envelope on some of this. And Steve is big into this, as I know some of you are as well. And then, you know, then the second part of your talk, I think, gets into the specifics of what that Jerry and I started a couple years ago. I mean, you know, and it's taken us a while to get a handle on this stuff. And Ari's been right in the middle of it, and we're just about ready to submit the first of about 5 papers that are all prepared, you know, and are going out 1, 2, 3 after so much work. It's like we're going to throw a party afterwards. Maybe we'll have it at the retreat. So anyway, Ari, go ahead and take it away, and you know, I'm sure that you know, leave room for people when they have questions, and let's see how it goes.
Ari, no questions.
Well, I'll see what I can do.
It's like the chalk talk. We won't interrupt you for 3 minutes. Okay, go ahead.
I'll do my best. So thank you everyone for coming. I'm sorry about the name. I know I didn't distribute the name for this talk until yesterday. The truth is I've had bad luck with names. I was working on a nucleosome positioning assay. I was going to call it Genome-Wide Assessment of Nucleosome Organization, and it turned out that's spelled guano. And
that's a good one. I remember that. Yeah. Don't get me going.
And then I decided to call this EpiGWAS, and I just looked it up. It turns out that's actually the name of a professional wrestler. So.
Really by the way, that might be a good name.
No.
But he doesn't pronounce it that way. So I want to talk about the influence of epigenomics on GWAS, the work that the AT Lab has done in the area, and where this is going. And the theme will be moving from association to predicted function with increasing levels of automation in the analysis. So to begin, I would like to talk first about the current state of GWAS, where it is right now, what a published GWAS paper looks like in the second quarter of 2015, and then transition into the feelings of people who are expert in this particular area, and a large part of this field is sitting right here in this room, about what the meaning is of our increasing understanding of epigenomics and the 4-dimensional structure of the genome, what that means for GWAS, and then talk about how these findings are going to be translated. And so to begin, I thought we'd get our feet wet and get some firm grounding by looking at a current paper. This is hasn't been published yet. It's coming out in about 2 weeks, Journal of Addiction Biology. This is a real live paper. This is what the field of GWAS looks like to someone who's reading a paper right now. So they've got a substance dependence phenotype with a cohort of patients. They've got people who are addicted to various drugs. They create phenotypes based on whether you're addicted to anything or quantitatively how many you're addicted to. They run a SNP panel on all these people. And what you get is a Manhattan plot that shows uncorrected p-values as a function of genomic position. You use a hard cutoff at a certain point, and you get out a certain small number of SNPs. In this case, a total of 4 SNPs. at a p-value threshold of 10 to the minus 6. And then you can take these SNPs and plot them one at a time, of course, because you only have a few of them because you've used this strict threshold, and start looking at odds ratios based on the genotype of an individual SNP for your phenotype. And you can see, oh, there's some results here. You then take the SNP and you associate it with a particular gene just by what it sits near sequence-wise, without any particular attempt to connect them functionally. And that gene may or may not have anything to do with it. And sometimes you're lucky to get a small number of results, because if you had 500 significant regions, then you wouldn't even be able to do this kind of low-level analysis. But then when you zoom in a little bit, you can see that, of course, it's not just one SNP, it's an entire linkage disequilibrium region that has a large number of different SNPs. And it's not necessarily clear which one or ones of these SNPs actually matters and which ones are just along for the ride with linkage disequilibrium. It's not clear how it works. It's not clear what the site of action is. And it's not always clear how to interpret the results that come out of this. And there's been a lot of emphasis within this field on applying epigenomic findings to GWAS, but I just want to make the point that at this particular moment, papers are being published right now that get basically zero insight from any of the stuff that we're discussing and are doing GWAS essentially the way it was first invented. So from the conclusion of this paper, you can see they're talking about genes. All of the variants are being described as genes. And in terms of what they could conclude from this paper, they can say, hey, there are variants that contribute to risk. This is this is somewhat genetic. No talk about what specifically those variants are. And then the results require replication to further explore whether the association is real at all. So this is kind of a weak and limited conclusion and very limited methods. And right now, GWAS is sort of at a crossroads in some sense. It's controversial even to say that the field has much value at all. And you can see some extreme opinions, like the blog of an evolutionary geneticist here who referred to GWAS as worthless gibberish, nonsensical hogwash, and a waste of money.
Which evolutionary geneticist was that?
You know, I looked him up and he wasn't that prominent. But
He wasn't the guy that said ENCODE is like a television.
I'm laying down the extreme ends of the spectrum. This is probably the most negative appraisal that you can get of GWAS as a field. But even if you look at a different blog, the CDC's blog, the impact of GWAS is being referred to as a glass half full or glass half empty scenario. The glass is not full and our cup is not overflowing, it's limited. And in specific systems, you have people looking at a large number of different GWAS studies in AthNA and going, okay, this wasn't that useful in this particular area. We've done a lot of these and it's not that useful. And you know, even if you look at the people who are defending GWAS, this paper from Human Genetics, for instance, 5 years of GWAS discovery, their goal was to defend GWAS and to say, hey, we've had wonderful results. But the first thing they had to do was include a summary of all the people who concluded that GWAS as a whole has been a failure. And in their conclusions, they talked about lengthening the time frame for understanding disease based on GWAS to decades, not years. So although some people believe in GWAS, some people don't, it's a controversial subject.
Well, this is excellent. However, I think we should acknowledge that some of the world's experts in GWAS are our collaborators and friends, like Mike Benke and Gonzalo Abacasis, and if there's been any contribution to the field of GWAS in the world, it's coming right out of here. And then I think another thing I would, which might play out as a theme later when if you show your smile plot, is that there's this whole GWAS, and Steve's been a part of Fusion for years. Looking for common disease variants, if you will. I mean, that's a certain area of GWAS study that has yielded some fruit, I would say. So, but this is actually very good to kind of show that there's some controversy out there. But you want to just give it a balanced approach, because in the end of the day, what you're going to show is that we're getting a lot out of it.
Oh yeah.
Okay.
Maybe all artifact, but
I don't want to belabor this
Get some duct tape.
either, but I mean, there should be noted too that, I mean, I don't know what, I didn't recognize the journal that you showed from that. And so, I mean, I think it's clear that there's lots of scientific studies that are published that are underpowered, that are maybe not as robust as others. If you were to look at modern GWAS studies in like, that are actually getting published in maybe more read journals, I don't want to say like the glamour journals, but even, you know, good journals, the bar is much higher for what you can publish nowadays, and they do have to have functional follow-up. And they
Well,
do have
what
to,
Kristen has done with cardiovascular, or the recent study with the meta-analysis of
The good papers are much better now.
340,000 GWAS hits that Speliotis et al. Published back.
there is a whole spectrum.
Absolutely
Yeah.
true. There
No,
are,
you're right.
There are better and worse GWAS, there's no question about it. And you know, the reason that I happen to run across this one is because if you search for GWAS on PubMed right now and sort by publication date, this is the most recent primary GWAS trial on PubMed, and it serves to illustrate that even if there's a
Previous
lot of-
slide, right?
Yeah.
By the way, it's out there. It's good to know about it. But, you know, Ryan,
It's like the old ones. They didn't even just say, oh, they end right at where they should be starting to actually investigate further, right?
So the point is that this mode of GWAS, although we don't like it, still exists in 20.
Fair enough.
Okay, good.
So in terms of GWAS, what you get out of this sort of static mode of analysis is linkage disequilibrium regions that potentially contain one or more variants associated with the trait. You get a lot of regions that have potential but questionable significance. If you look at that Manhattan plot, there's huge chunks of the genome that are 10 to the minus 4, 10 to the minus 5 p-values, and we've been trained to ignore them because there's no hope of interpreting them on that static basis.
It's an important point to make.
These incredibly strict, hard cutoffs to get down to a small set of variants, which are then still difficult to interpret. Because people love coding variants and tend to ignore, at least to a certain extent, non-coding variants, you've got a large portion of the lead SNPs being somewhat ignored. And there's this missing heritability problem. And instead, what we want out of GWAS is to get particular variants, not just LD regions, that are causative. We want to have some kind of information about how they work and what convinces us that they're causative, and we want to capture more of the heritability. And so this is what the future of GWAS is going to be working on, whether there is a future or not. I want to talk about non-coding SNPs. These are crucial. As Dr. Boyle pointed out in his 2012 publication, 95% of reported GWAS lead SNPs are non-coding. But if you look in PubMed and try to find publications talking about non-coding GWAS, whether you search in all fields or in just the title and abstract, only 1% of GWAS publications even mention non-coding anything.
Wow.
So, you know, as Philoso, Raptor, and college liberal protester tell us, there's a disconnect here between the literature and what's going on in the results. And part of the reason for this, I believe, is that non-synonymous coding SNPs are very easy to interpret. You say, oh, this protein gets changed to a different amino acid, does something different, it causes the phenotype. Whereas non-coding SNPs are, of course, a lot harder to interpret, at least facially.
Well, a lot of things were done as reference SNPs that were associated, and if they didn't see anything, they just report the reference SNP.
Right.
It wasn't so.
Yeah.
They're in the literature, but they just didn't know where to go.
Right. They get reported but not talked about, which is
Well,
problematic.
They didn't do any analysis.
As far as
that
any
bit-
mechanistic interpretation.
That figure down below is kind of telegraphing everything, right? I mean, because that's saying, hey, you know, ENCODE and epigenome roadmap happen. Patient stratification is real and can add power. I mean, everything that's kind of the future is kind of summarized down there, and I guess that's where you're taking them.
So then you've also got the question of missing heritability. You have these heritability estimates that come out of family, you know, pedigree analysis for these diseases, and then you look at GWAS and you start adding up the effect sizes of all the SNPs that you get out of some GWAS, and you find that there's a lot of heritability that you're not capturing. You might have a 60% heritable disease, and the GWAS variants that you can find only add up to 3%. And there's controversy about what's causing this, where the missing heritability is to be found. You know, some people think it's from pathologically large numbers of very small effect size variants. Find. There's belief in direct epigenetic inheritance controlling a lot of this stuff. And there's also the question of combinatoric interactions between variants that are not being captured in marginal effect sizes of single variants.
Right.
And of course, combinatoric analysis is prohibitive for genome-wide variant sets, because if you have 1 million SNPs and then you want to look at sets of 3 SNPs, you now have about 1 quintillion 3 sets to analyze, and the cohort size you need to even approach statistical power for sets of 3 would be really troublesome.
World populations.
Yeah, there we go. That's approximately what you would need. And so if you really want to do combinatoric analysis, you need to find the high-quality candidate variants that might be participating in these high-powered combinatoric subsets. And so the missing inheritance problem is sort of an element of what's going on in GWAS, that it's not revealing the missing inheritance. So in terms of epigenomics, what's been happening since GWAS was invented and while these studies have been ongoing is that we're now having an increasing understanding of what the functional elements of the genome are and how they are both demarcated and to some extent controlled by epigenomic marks. And to some extent, we have evidence of direct explanation of heritable phenotypes by epigenomic marks. We also have increasing availability of data on these marks in high-throughput genome-wide data sets for a lot of different tissues and a lot of different assays. So here are the data grids from both the epigenome roadmap and from ENCODE, showing with data types on the top and cell lines on the left, how you can, to a certain extent, particularly for highly populated data types, pick through your cell lines and tissues and pick data types of your choice, and genome-wide data sets are already there, so that to a certain extent the epigenome is now something you can consult, as versus having to necessarily generate new data for every experiment. And so this has been getting increasing use, as we've talked about, both in interpreting GWAS results and annotating variants on a static basis, on a genome-wide basis.
I think that's Steve, I'd be curious about your interpretation of what he just said, but it seems to me that some of those epigenomic roadmap papers, at least, work this way in spirit.
Yeah, I think that's great. I think you had to have and maybe I didn't hear this, but you had to have the disease-relevant tissue. That's critical to interpreting.
Yeah. Yeah.
And that's
Absolutely.
a pretty hard substitute.
Yep.
And the other thing that's been going on the last 5 years is the increasing understanding of what's going on inside the nucleus in 3 and arguably 4 dimensions, where we now understand more than ever before about how the genome is organized and what implications this has both on nucleus-wide levels and on relatively small levels for the control of transcription and how that's influenced by variants. And this is a field of intense interest. There's been a proliferation of data and an ongoing proliferation of data that will continue, including the development of reference cell line and tissue collections analogous to the Roadmap project and ENCODE project of high throughput ligation sequencing, high-C, for different genomes. And in addition, there's a proliferation of other assays for similar information. We've seen data from both 5C and ChiaPet.
But yesterday we saw 4C, in the lecture, and I think that was very effective.
Right.
Yeah.
4C is not necessarily going to be that useful for doing GWAS analysis, simply because it has a target locus. And so to do genome-wide sets.
No, you use the GWAS as the target.
Yeah, right. You use whatever you found, right?
Yes, if you're willing to.
Yeah.
But in terms of using it to screen variants on a high throughput basis, you can only do that for a small set of loci after you've already taken them out as the results.
The GWAS loci.
Yeah, that's the point.
Yeah.
Use that as your,
It's the bait.
Right,
the
that's the
bait.
bait, right.
Yeah.
Like he was showing, right?
Sure. Once you're going into very deep analysis of a very small set of variants
Yeah.
that is coming out of the experiment.
Exactly, which is, the way you might want to.
Yeah.
Certainly would fit with our research that might be exactly what you'd like to do.
Yeah.
Yeah, so how do you explain genome architecture map?
So there's
I shouldn't even say that.
There's another
You can
technique
just
that
go
that's
on if you
okay,
want.
don't worry. There's another technique for getting access to genome spatial information that's just recently been developed and hasn't been published yet, entitled genome architecture mapping. That's based on correlation analysis of the appearance of different sequences in the genome together in single cell genome sequencing of thin slices through the nucleus. So the notion is that sequences that statistically are more likely to appear together in those single slice genomes are statistically more likely to be to have close physical distance. And one of the things that's exciting about this is that it operates on smaller numbers of cells, and in addition has the potential to go from interaction frequencies to actual physical distances, assuming a good spatial model of the shape of the genome. So we can talk more about that.
I would think as you go through the nucleus, you'd end up sort of in the middle, and it'd be like things that are far distance apart are going to be coming out.
Yes, things that are far distances apart definitely do appear, but they do not have as strong of a statistical tendency to appear together. So there's a relationship between correlation coefficient, between pairs of loci.
Does that suggest that the nuclei have different orientation, or the chromosomes in the nuclei have different orientations from cell to cell?
Yes, definitely. And translating these results into physical distances are going to require some good modeling, a better understanding of nuclear shape and its relationship with the slicing. But this is probably a derailment relative to the main topic of the talk, and I think we should probably move on. But yes,
Yeah, I'm sorry.
We should definitely
I
have a
derailed.
talk about GAM in the future. So increasing amounts of nucleome data. And we're learning a lot about what goes on spatially inside the nucleus from this. We've learned, this has been known for about 15 years, that individual chromosomes live in these distinct chromosome territories that do not overlap very much, that chromatin is divided into multiple compartments with different activity states and replication timing, and also histone marks, and that, and this is just the last 4 years, that chromatin is organized into topologically associating domains. In the last few months, we found that they are loop and ordinary domains, and that loop domains joining promoters and enhancers are associated with transcription. And so we're learning a lot more about how the genome is organized spatially. And what this means in terms of GWAS is that we're beginning to develop a picture of where transcription lives. That transcription lives near relevant enhancers spatially, in loop domains, in transcription factories, in the open chromatin compartment, at the boundaries of chromosome territories, in the central portion of the nucleus. And so in general, if we believe that we've found a locus that functions as an enhancer regulatory element for a particular gene, spatially, in terms of spatial interaction data, both Hi-C and FISH imaging, the more of this profile you can fit for your individual loci, the more evidence you have that that's how it's functioning.
So, Ari, just a minor, I mean, this is almost turning into a religion for us in our lab, but I mean, is this a hypothesis that we're going to test? I mean, we hypothesize that, these non-coding variants live here, live there. I mean, what's the evidence?
Sure, this is definitely testable, and to a certain extent is being tested and has been tested by a certain amount of literature. There's going to be, a greater amount of testing of this picture on an ongoing basis. What you would do in order to look at it would be take systems in which you know that a particular gene is or is not being controlled by the action of an individual enhancer.
Yeah,
And then compare those two conditions with respect to all of these things.
I don't know if you're going to show it, but we have these diagrams that we created, these circus kind of diagrams that show putative cis or trans relationships that could be looked at directly through imaging. That could lead you in the direction of validating these in the appropriate cells and tissues.
Definitely.
Using the microscopy.
And a certain amount of this imaging has already taken place in a lot of the seminal.
That Rao et al. Paper had that one figure 7 or whatever.
And just to look at the first 2 bullet points here, spatial contact with enhancers and loop domains, multiple different Hi-C papers have shown a strong association between spatial contact between enhancers and genes and the level of transcription of those genes in different cell types, comparing cell types or conditions. And also, of course, Rao et al. Showed that the presence of a loop domain in the body of a particular gene was strongly predictive of its transcriptional status. So there is some evidence for this. It's not just a religion.
I see. So, but maybe this slide came a little early because what you're going to show with the pipeline is the for a system of our interest, non-coding variants that are assigned to RS numbers that are in,
yes,
associated with enhancers, the way Steve found with his stretch enhancers and all that. And then once we have those, then we would go and look.
Yes, the results
Yeah,
I'm going to
okay.
show are concordant with this.
All right, okay.
So what's been going on over the last few years with increasing involvement from me is this imputation pipeline, which I'm calling the Higgins et al. Pipeline. Jerry tells me he doesn't want his name on it, so we'll call it the Allyn-Smithy pipeline.
Yeah, right.
But,
It's something we've been working on. It's getting better.
but so it's a workflow, and this is mostly manual at this particular moment. There's a certain amount of scripting, but a lot of stuff depends on spreadsheets being copied and pasted by humans and humans consulting genome browsers. Starting with published GWAS results, assemble a broad set of candidates that are within a linkage disequilibrium distance of genome-wide significant lead SNPs, score them with multiple forms of epigenetic information to find putative functional SNPs, with the goal of finishing with a smaller list of higher quality candidates that have predicted meaning and function that can then be tested in downstream analysis. And so we'll talk about the method, what this does, the results, and then conclude with our plans for expanding it. So start with a permissive set of variants starting with lead SNPs and extending to all the SNPs in linkage disequilibrium. Currently, we're using the traditional genome-wide significance threshold, which approximates a Bonferroni multiple comparison correction on a genome-wide basis. But in the long run, like the rest of the GWAS field, we want to transition to false discovery rate methods to do the initial screening.
You can use other tests too. You know,
Yes.
Fisher's exact. I mean,
And one of the things is that, in general, the more strict is going to be the screen that goes on, and the more automated the procedure is, the more permissive you can be with the initial screening. And we'll talk about that a little bit later. So the variants are scored with a weighted combination of various forms of epigenomic information, including QTL predictions, chromatin accessibility, methylation, histone marks, transcription factor binding site measurement, and bioinformatics, and Hi-C mapping. And this is very similar to the work of Boyle et al. We use data sets so far as is possible from phenotype relevant cell types, whereas at least the publications that I read from Dr. Boyle were mostly...
It's too bad he's not here right now, but
yes.
we've tried our best to give weighting terms that were reasonable, which were, frankly, an extension of what Alan had, you know, in his earlier paper, which were all kind of qualitative check.
yeah.
And there's
But we really are looking for, you know, kind of a thesis project to get a better scoring function, especially when we add the Hi-C or some of the other things. We really think that there's a need for a, you know, biomathematical statistical scoring function here, and we're not pretending that we have one.
right.
Right? I mean, it's a huge need.
One of the differences with the regulome DB work is that rather than attempting to compile the human regulome at the greatest possible depth, what we're trying to do is find the regulatory interactions for a particular set of variants in a particular system. So we're using, rather than the most evidence cell types that have the most evidence available, we're using cell types that are as relevant to the phenotype as possible.
Yeah, so we should be clear here, because, you know, this pipeline could be used, you know, I think generally. But you can see, you know, I don't know, maybe I missed it, Ari, but, you know, our motivation for doing this was that we were interested in pharmacogenomic response and extending the idea of that beyond the exome. And so, you know, we just downloaded, you know, data from the GRI catalog and psychiatric association studies that were related to, you know, neuropsychiatric disease generally, that would include even things like epilepsy and stuff like that. So, and when we were looking at the, these were all pharmacogenomic studies, so we were looking at, you know, how people reacted to the drugs and whether there was any adverse drug reactions and things like this. So that's why, you know, and the kind of the clue is, you know, that thing down there, RNA location. I mean, is it in the liver? Is it in the brain? You know, is it a pharmacokinetic response or pharmacodynamic response?
And obviously, those are going to have to be tissue specific. So if you were
Yeah.
investigating, you know, skin cancer, you would want skin tissue.
Do you have
Right.
alternative ones of those?
Sorry?
Is that the pathway, or are there alternative ones that are used? My question specifically is why hypomethylated required? It kind of presumes a certain mechanism of action.
Right. This is what we're currently using. It's definitely true
Yeah, but
that if you're looking at...
You could turn some things off that are regulatory and would influence a whole bunch.
Right.
Yeah.
If you're looking at variants
So-
with different modes of action, then what their statuses would be different.
And that's just two quick things. You're probably going to say this. I mean, that really is based on the regulome work out of Mike Snyder's lab that Alan Boyle and others have done. So we have to give them, well, we already have appropriate kudos. The other thing is that
Edwards.
yeah, Edwards, right, who I work with at Coldstream Harbor. But also, everything we've found, with I think one exception to date, is confirmed by, if you believe it, the Epigenome Roadmap Consortium.
Well, it's a slightly separate question. I believe that
Yeah, it is.
Have-
Yeah,
Or,
not
or,
really
is it,
answering.
or is it the way the variant results in hypomethylation? Is that the way I should?
no.
Or
No,
is
this
it
is just a-
hypomethylation
this is just
place?
that the
this is
locus
just
where the
an
variant
annotation.
is.
You know what I would-
hypercorrelated with-
You know, it's a good catch. You know, and Jeff's really, he's part of our lab, and he makes catches like this all the time, and I actually like it.
We fight all the time.
I think, well, and we do fight once in a while, but, you know, I think state of methylation state would be better there.
Okay. No, and I do
You know, we
actually
still have
a
time to
slide.
change that. I don't see how that would hurt us right now.
Well, I mean, basically, what you're pointing out is that what we're assembling here is a profile of what a functional variant looks like. And variants with different functions may have different epigenetic
But,
profiles.
you know, I think we just
And
need
so
to,
and so I think that eventually what we will want to do, and I discuss this in a slide later in this presentation, is break the scoring function out by functional class, predicted functional class of the locus. Because if you have a locus
Fair.
that is supposed to be a promoter, it's not going to have the same
But-
set of.
by the way, that's fair. I mean, you know, I simply want to say that this workflow is really tuned to neuropsychiatric, so it's mood disorder, so biochronicity status makes a difference.
Yeah.
You know, all this is a pharmacogenomic, you know, neuropsychiatric, you know, workflow, and what we should really think about is generalizing this at some point to a more general workflow, and that would be worth doing.
The subject of the later part of
Yeah,
the
that would be worth doing.
So, and so then the next step after the scoring is to take
good.
these variants and supplement the epigenomic annotation with chromatin state modeling and also with Hi-C or ChIA-PET 3D interaction information, and then to take these the variants that pass through these additional screens and compare them with obviously known pathways that are involved in the diseases, because now we have genes that are associated with them through EQTL relationships, so you can start looking at what those genes do and whether those are known sections. And so if things go well, you finish this pathway with SNPs that have not just an association, which of course they do have, but also a predicted function. This is an enhancer that modifies the transcription of a particular known gene, things that could be really testable and give focus to subsequent work.
All right, this is cool. So this, just to talk about it later, but this ought to actually be figure one, if you think about it, because it goes from beginning to end. Yeah, I mean, this is really the pipeline. You got the GWAS stuff, you got the you know, kind of the imputation stuff, then the mapping stuff, and then the validation stuff, you know, extension 3D stuff, and then the validation. This is actually the pipeline.
I believe this is
Yeah,
figure one.
but it is in the paper,
Okay,
Brian.
good. So this is now
I-
figure one, good.
Yeah. Yeah, it is. Yeah.
Excellent.
So that's approximately what we're doing, and so I'd like to talk about some results that have come out of this. So this is going to be largely based on a paper that we're submitting tomorrow to Applied and Translational Genomics. So we took 28 GWAS studies of psychotropic drug-related phenotypes, including addiction, which this catalog of studies doesn't include the one that's going to be published 2 weeks from now. So this is perhaps an example of what that study would look like if subjected to additional analysis on the basis of epigenomics. Analgesia with opiates, drug response for lithium to depression and bipolar, I believe, and then adverse events and organ injury associated with drugs. And then analyze these results with this pipeline. So you can see here the table of 28 GWAS studies that went into this. And the results are very much in terms of the way the SNPs pan out. We've got about 2,000 SNPs that are either lead SNPs or linkage disequilibrium with them. Only a very small percentage are coding variants, 6%, about the same as was discovered by Boyle et al. Most of them
And
sit
UCAS.
either in introns or in untranslated regions of genes. And among the ones that come out of the workflow, you can see that the bulk of them are predicted in terms of their chromatin state to be enhancers and promoters. Transcribed genes are a small fraction, and among the transcribed regions, they are, I believe, all three of these are synonymous. So the non-synonymous transcribed mutation is not a big thing in the results that come out of this. And you can see here the chromatin state annotation of the 30 SNPs that came out of this workflow. And you can see the chromatin state annotations that were predicted by the workflow are in very good concordance with the ones that came out of the epigenome roadmap predictions data. And these are coming from different data sets and different annotation methods.
Is this for a single tissue type, or?
Yes, these are for the relevant tissue types for the disease, which in the case of all of these, except for, I believe, the addiction ones, for alcohol and nicotine. Or,
You
sorry,
can see
Oh,
the
I see on the
tissue,
right.
so it's,
Okay.
so it's either liver or brain, which was that thing. But you'll notice that that one does pick up in the pancreas, which I thought was weird, and we have some CD14 cells.
Yeah, actually, and we have it split up by different brain regions too. If anyone's really into the
Yeah, that's
phrenology
another
of
paper.
neuroanatomy, that's another whole thing.
Yeah.
There were rewards.
Yeah, we actually go into the brain and using Allen Brain look at the transcriptomic maps. That's a whole other paper. It's circuit level analysis.
Right.
Okay.
So here you can see a summary of the transcription factor binding coming out of ENCODE data, I believe.
That's not, and by the way, that's not all of
That's
them.
not brain.
Sorry?
That's not brain then.
Oh, am I wrong about the data source, Jerry?
Oh, this is, oh,
This is totally different. No, this is. Go ahead, Jim.
yeah, okay, so this is multiple tissues because, remember, some of this is liver, some of it is
Right.
actually pancreas, I don't know if you caught that, heart,
But the
immune
bulk of it is
cells, and brain. So it's a combination of different things. And these are not all the transcription factors.
No,
It's
of course not.
So there are other papers, and I mentioned this, and I'll shut up, but there are other papers that have shown, depending on cell types, certain transcription factors are highly significantly associated, as you might expect, and maybe people know, with enhancers and or promoters or transcriptional start sites, and mechanistically shown, right? I mean, you can sort of extend the notion of a transcription factor to include things like EP300 or, you know, so what I'm saying is, we were trying to pick things that could define, I don't know, should I shut up?
No.
One things I wanted to do is see.
Promoter enhancer. You know, this is really
Yeah,
the
so
3D
I wanted to
stuff.
see what might be near boundaries.
Yeah,
So
that's what we were
defining-
interested in.
Right, things that were close to CTCF or cohesin, which would then be RAD21 and SMC3, if that makes sense. Or this is all inference, right? But it suggests that it's close to what I used to call an insulator boundary, but I don't think that term's even used by people.
It's an old term.
Yeah, it's an encode kind of term.
Yeah.
But what we liked was the binning on addiction, and still have to change pain to analgesia, but you know, those categories, which are the same categories as the first slide in that we're following.
Right. Plotting these variant. Oh,
Well, this is important stuff here. Go ahead.
so in addition to the concordance that we saw on the last slide between the transcription factor binding site changes that were changing motifs in response to these SNPs, being concordant with the transcription factor binding that was measured by ChIP-seq, there's also a concordance between the EQTL predictions and spatial connections between the SNP loci and the predicted promoter TSS loci for the genes for which they were predicted to modify transcription. And so this is, of course, consistent with our belief that promoter enhancer spatial contact is one of the key drivers of transcription, circa the Rao results and other recent HiC results.
So it's a shame, though, that, you know, those things are so small that you can't see them. But, you know, we can take A up there in the corner. And so this is cytochrome CYP1A1 and CYP1A2. And those little black lines show the position of the SNP within the, you know, the loop, if you will, of those genes.
Right, within
And-
the enhancer, actually, that's
Exactly, and the
defined by some
enhancer
other
is
source.
Right, exactly.
These things are, by the way, obviously not, their own, it's very qualitative. They're not to scale, right?
Right.
So,
Stylized representation.
yeah, there's very stylized.
But I lost, darn it, the printout. Oh, here it is.
Otherwise,
I mean,
this enhancer would cover 3 chromosomes.
but the cool thing is that so those CYP genes are on chromosome 15, but that trans interaction what we're describing is on chromosome 9.
Right.
And so what we're trying to do is come up with some kind of way to indicate, kind of the circle of influence, if you will, that a certain SNP could have in 3D to other important genes that are not, contiguously connected, but are close in 3D space. Is that a fair statement? So, we could go through each and every one of these, but and Steve, you might, have you seen plots like this before?
Yeah, so maybe I don't follow this. So the red line inside the circle in A is connecting an enhancer on one chromosome.
Represents a spatial interaction in high-C data between 2 portions of the genome.
But it's trans, right?
It's trans, yep.
Okay, the first one's trans. It's like, what is it? A, C, what's that one? E down there?
That's cis.
That's cis.
And
Those 2 on the bottom are cis.
And also
Those 4 up there are trans.
so the trans ones, those are hard. I feel like they're really hard to detect.
Yes.
I don't know how you're doing. Are you doing it just with high C data?
Yes.
Okay.
Relative to background, but yes, it's tough.
And it's not on the relevant cell type either, right? Because you probably don't have that for brain or liver.
I believe it was a brain cancer cell line, which is brain, but it's...
Or if it's CYP, it's probably HepG2 or
It's
something
liver,
like that, right?
HepG2.
Okay.
So what we should do, what I'd like to do, Steve, is we could talk with you about
Okay.
it.
But can I just say something?
Yeah.
What is bizarre about some of these is
What they're connected to.
yeah, they're connected to genes or a gene on another chromosome, and I don't know where in high-C data that explain
Like this
the
CAG
mechanism.
A1B, for example, and figure C down there. I mean, when we see it, and this is actually this other paper that we're just working on now, when we see it, the connections are like saying, oh, we know that gene, that's related to this mood disorder or this drug response. It's not like it's just some kind of random gene that we'd never heard before. And so what we're doing now is that, we're doing pathway analysis to try to understand that. So we have the GWAS over here in the pathway analysis, and that's already, kind of showing some new things for us. So there's kind of 3 levels in the thing. In the brain, there's 3 levels. There's the GWAS SNP thing with these kind of inferences. Then there would be the pathway analysis, and then there would be ultimately how it works on a circuit. Does it map to a circuit that is indicative of a certain disease or not? And that's from a pharmacogenomic perspective.
So, can I just give an example? Like in C, and we can map these better. These are pretty crude things, by the way, that I did manually, as you could tell. We tried to use Circos, but it becomes a mess. What's really weird, this is one of the best examples, and Brian can vouch for this. We found the spatial connection in C between patients in a GWAS that had prolonged QT syndrome, if you guys know what this is.
How many people know prolonged QT syndrome? This is really pretty important. Okay, we're getting a bigger thought.
No cardiologist
So
in
this
this
is
room.
really, I was just on the phone with the FDA before, just coming in the room on this. Tyrosine kinase inhibitors, which you use to treat cancer. What's the big adverse event? Prolonged QT syndrome.
Right.
People are dropping off with heart failure.
Yeah.
So a certain number of these TKIs, cannot be used, especially with certain patients, because they would be susceptible to getting this heart defect. We have this with, antidepressants.
Right.
Some antidepressants show a strong correlation with this prolonged QT syndrome, and it turns out it's a big, serious adverse event. you know, people quit taking the drugs, or, you know, we try to find the marker for that so we can prevent them from taking the drugs, that kind of thing.
Yeah, so, and see, and Brian, you can, and I'm sorry, Ari, but I'm proud of this, but it could be coincidence. We found this, so quetiapine is also known as Seroquel. It's a atypical antipsychotic, and there's a SNP that's highly, in the GWAS replicated, associated with prolonged QT syndrome.
Yep.
It's a non-coding SNP. It detects it in the middle of a known enhancer, at least described by other people, not necessarily functionally characterized, right? And we mapped it to, using high-C data, to this other gene, nitric oxide adapter protein 1. Since we've mapped this, there have been almost 20 publications that have
20 pubs that are showing us
that say that that gene we mapped it to is controls, so mutations in it disrupt QT prolongation, and it actually controls in heart and cardiac muscle the QT wave. So is that coincidence? I don't know, man. I mean, you guys tell me, right?
Brian and Jerry, I don't want to trouble you too much, but we have at this point 19 minutes.
Good.
Brian, Jerry,
Go.
19 minutes left.
I'm sorry.
Go. So we dove in because this is our passion right here on the right.
Because we're finding so many things that explain mechanisms.
So go ahead.
It's 9 minutes.
Steve, you got to
Well,
tell me, is this real?
I mean, this is an hour-long talk, right? Oh.
Goes to 1.
Right.
All right, let him talk about this. This is our piece de resistance.
Thank you. So, independent of the sort of interesting and suggestive pattern of what the genes are that these SNPs have QTL relationships with, they also have a significant effect on the phenotype, not just statistically significant, but clinically potentially meaningful. So this is a plot of effect size on the y-axis versus allele frequency on the x-axis. The effect size is calculated from odds ratios with Cohen's d-test. And this is a plot that's come up in GWAS analysis, particularly meta-analysis, several times in recent years. The plotted lines are statistical power curves representing the statistical power threshold of a GWAS study. In this case, they are hypothetical GWAS studies. The gray dots on the bottom represent risk SNPs for disease risk of some of the syndromes,
Psychiatric disorders.
And the power lines represent the statistical power in black of the median disease risk SNP, a typical disease risk SNP. And the top red line represents the statistical power of the GWAS that would be minimally able to detect the strongest disease SNP. And you can see that 80% of the SNPs that have available odds ratio data that are coming out of our imputation pipeline are sitting above the statistical power of even the strongest disease risk SNPs. And this is striking because in certain circles, there's an emerging belief that risk SNPs are governed evolutionarily by the negative selection effect of the negative fitness of a disease or other undesirable phenotype to have a statistical ceiling on their combined frequency and effect size, whereas we find that the putative ceiling for disease risk SNPs is not being observed, which points to the fact that GWAS may be fundamentally different in how useful it's going to be for us, and the landscape of variation controlling phenotypes may be totally different for different phenotypes. And you could sort of put on your evolutionary biologist hat and start spinning stories about how addiction to a particular drug, susceptibility to addiction or drug response might not have been evolved for during the primordial period because there was no lithium supplementation during that period. But then on the other hand, you do have drugs that humans have been using for a very long time. So the evolutionary interpretation is questionable, but what I think is pretty strongly evidenced is that the availability of these highly desirable, high effect, frequently observed alleles varies significantly by system, and that these pharmacogenomic systems do appear to have more availability of good variants.
Can I just add something? This is the first time it's really been shown for psychiatric pharmacogenomics or neuropsychiatric, including neurology. But other people have shown this for other pharmacogenomic SNPs. Right. That there are not only quantitatively, but qualitatively different than, I don't know if you call bipolar disorder a complex disease or not, but then what we would call is common disease or complex disease SNPs, if that makes sense.
Right. So this we refer to as the smile plot. So this is one of the most exciting results that comes out of these, because not only are we sort of spinning these stories about what genes are associated and what involvement they have, but these also appear to be really good variants in terms of their predictive power. So just to summarize what came out of this, instead of just association with a huge cluster of SNPs and no knowledge of which one matters, we start getting specific SNPs that are not only associated but also have predicted chromatin state with concordant epigenetics and transcription factor binding, a predicted gene target with some evidence that they're linked spatially, and in some particular systems, including this one, common SNPs with large effect sizes. And in particular, sometimes we're finding SNPs that traditional GWAS analysis didn't mention, that were in the LD cluster but were not specifically called out by the source studies.
You had common? Well, I guess it depends on what you define as common.
Well, 40%, 35%.
These are all population stratified, by the way.
Right.
What, that's not minor allele frequency. That's like derived or something, right?
Yeah, deja vu all over again. No, go ahead, Steve.
That must be derived allele frequency, I'm assuming.
Sorry.
What allele frequency are you plotting there?
The allele frequency of
this
the
is from specific populations,
Right.
or like very small populations.
well, the population.
No.
The
Well,
populations
you go on.
No, if you
you're
look
getting
at-
it from a
okay.
stratified, a small, a relatively small.
Yeah, no, I thought we were gonna change that.
frequency
I
within.
thought we were gonna change that axis down there
Yeah, it bothered me that it was.
to.
minor allels to me.
So
It's not minor allele frequency. It can't be.
No, it's not minor at all. It's
It
up
is
to
in the
one,
world.
man.
the
it's
frequency within the
but
within
it's
the
in the-
relevant
it's
population. of the
stratified by
stratified. It's a stratified.
haplotypes, just as a general statement, are different based on ethnicity. And anyone that says that's not true, I don't think really
Well,
understands
listen, all our
population.
data is
That's not
indicated.
the argument, but yeah.
We're not arguing that.
No. The point is that these frequencies are coming from relevant populations. It's not a global frequency.
So what should we call it? That's. And let Steve answer.
I don't know. I had to think about it.
Let's think. Do me a favor.
Right, maybe Ryan.
Yeah, I'm trying to think how you would. I mean, this is the. Yeah, I don't know how you'd phrase this.
Yeah, I had to think about it too.
Guys, do me a favor and think about it, okay? Because we've been kind of going back and forth on this one. What's that? Did you describe those way up high ones?
Sorry. Oh, this is a cluster of SNPs associated with organ injury in response to particular drug.
So how many people know about Steven Johnson syndrome? Have you ever heard of that one? Boy, besides you, this is a pretty important syndrome, and it comes up when you take a certain drug, clopazamine, carbamazepine. This is a drug that's often given to folks with epilepsy.
Right, and also bipolar disorder.
And also bipolar, yeah, oh yeah. And, there is a very severe rash that can ultimately kill you. That is a side effect of taking this drug that certain individuals can suffer from, if they have these markers.
Yeah, and by the way, can I just add?
Yeah.
Now Taiwan has come on board. So the Singapore and Taiwanese governments have
So Taiwan is now on board?
require those three up on the left there, that every patient be genotyped for those. It's law.
Before you take the drug.
Before you take the
So this came out in Genome Web about 6 weeks ago or 2 months ago, that, this is just such a, in the psychiatric field and in the epilepsy, neuroepilepsy field, this is a well-known syndrome.
Right.
But, the key is that you need a marker for this that you can give to the patient before you take the drug. And so this is a case where, the allele frequency isn't much, but look at the effect size. And it is huge. Now, the thing that, I'm still going back and forth with Jerry on, which fascinates me, actually,
Hey, Brian,
is...
I need to keep moving.
All right, but I just want to hang on a second, because I want you to describe this data. You know, you got these clusters here, and they're all telling a story. So if you don't tell the story, I'm going to tell the story. That's all. So, you know, because those other clusters, you know, are just as informative. But go on.
Thank you.
So, this is like, you know, it's like kids breaking away from the nest. It will happen. Go ahead.
Well, I'm not troubled. It's just that I think people are going to start leaving.
Yeah.
So instead of just association, we're starting to get out information that's a lot more useful and a lot more valuable. And in addition, sometimes finding SNPs that the traditional GWAS analysis in the original papers didn't pick out. And more of this is coming. This is the first publication that touches on a number. Yeah, I had Billy Mays there, and Brian said I needed someone more reputable. Jerry thought it looked too much like him when it was Billy Mays. So. But we have papers coming on the response to lithium and citalopram, and whereas these results were based on analyzing a relatively small number of papers per phenotype, in lithium and citalopram we go deeper because there are more GWASs available for the same phenotypes. And find larger numbers of genes and associated variants, and start doing some pathway and gene set enrichment analysis with gene sets from the output that show both replication of some existing pathways and also some new undiscovered stuff. So there's more of this coming, and you'll see more of it on an ongoing basis. So now I'd like to talk about some of the limitations of this process. It's largely manual. There's a number of, if you want to start executing that workflow, a lot of the steps involve looking up some genome browser and typing in loci one by one, including, GUI-based software packages and pushing data back and forth in spreadsheets. It relies on the source GWAS studies for this hard p-value cutoff and other data on the SNPs, for instance, odds ratios. And this definition of pharmacogenomic SNPs, this weighting that we apply to the different systems, I perhaps little more than guesswork is a little harsh.
No, it is. It's just guesswork.
Moderately more than guesswork, perhaps. And it's also phenotype specific, linear weighting with intuitive weights.
Yeah,
There's no
we need a model.
combinatorics going on. There's,
Yep.
Right. And so what I'm currently working on, what we're currently working on, is a new package for analysis of GWAS that takes this workflow, automates it as much as possible, and also improves it significantly. So, instead of starting to use genome browsers, we want to draw our data from locally stored genome-wide data sets or via APIs, pulling the SNPs out of scripts so that this data starts getting collected and used on an automated basis. We want to start with a permissive threshold because now that things are automated, you have the ability to analyze a larger number of SNPs and put more of the selection into the epigenomics rather than the GWAS p-values.
That's a good point. And the pathways.
And so this is sort of a co-imputation strategy wherein the p-value is not used as a hard cutoff prior to epigenomic imputation. They're being applied together, collecting all these different data sets. And then we want to have a scoring system that, rather than being linear and intuitive, is potentially trained on non-functional variants, or at the very least is based on a more systematic understanding of the comparative significance of these things globally. And that has potentially a tolerance for missing information because we're still drawing data sets from published work rather than generating every single piece of data ourselves. So sometimes the information will be missing. How many transcription factors have binding information for a particular phenotype relevant tissue is not going to be consistent. So in terms of the workflow, you'd start with GWASs in a phenotype of interest, can come from the literature or they can be yours. You assemble genome-wide data sets, including epigenome and Hi-C data and ChIP-seq, in relevant tissue and cell lines, where relevant is defined by the investigator with reference to both relevance and data availability. And then make a master run file specifying where the GWAS data is coming from and the data inputs. The scoring system on a variant basis will start with scores for each data type for each SNP and produce a figure of merit as predicted. The method, as we talked about earlier, could break into cases depending on the predicted variant type. So if the variant is predicted to be in a promoter region, then precisely what we want it to look like if it's to be predicted as a functional variant is going to differ. the cis and trans interactions, whether the interaction with its QTL targets are cis or trans, can be an important possibility, so we may want to break it into cases. And this might be created on a knowledge basis, or if I can figure out a good way to do this, potentially could be trained with machine learning on a known set of functional variants. And the limited reading I've done suggests it may be difficult to find such a database that would be unambiguous. So this may be a pipe dream on my part, but it's what I would like to do. But in either case, the scoring system should be a part of the system, not a thing that the end user has to come up with. Yeah. And this would be followed, limited, of course, by sample size, by combinatoric evaluation of effect size for small variant sets, because once you have a small set of variants, you can potentially do combinatoric effect size. So the user would be selecting a phenotype GWAS studies in tissue, collecting the literature data and or performing experiments of their own design, running the workflow, and then receiving not just associated variants, but predicted functional variants with information about their functional class, predicted site of action, and a summary in spreadsheet form of the evidence for this function call. And so I think that this could be something that GWAS investigators could use, not just the people who are writing it. And so if widely deployed, this would potentially change the meaning of GWAS, because you'd have a tool that would be usable by many researchers to get access to this kind of information. You would get more actionable insight from GWAS results, not just ongoing future GWASs, but also the past catalog can start to be reinvestigated. And you might start finding more clinically useful variants, and you might start having more understanding of what the target genes are for ongoing research in particular systems. And in general, we would hope to get results that are not, as we called it earlier, worthless gibberish, nonsensical hogwash, and a waste of money. So just to go back to what we talked about, we spoke about the current state of GWAS and how it is marred by limited meaningfulness of the results, how there are people out there who think it's useless, and even its defenders are forced to admit that GWAS has not necessarily panned out the way that it was initially expected to, how epigenomics and the 4D genome are enhancing our understanding of the function and how they should be applied in interpreting GWAS. We talked about the Higgins et al., the Allyn-Smithy SNP imputation pipeline, which does integrative screening of GWAS results and some of the results that have come out of that. And then we spoke briefly about my plans for an EpiGWAS package to improve and automate this method for use by other researchers. So thank you very much for coming. Thank you for your time and attention, and I will take
Very much.
So the x-axis should be associated allele frequency. I'm assuming because you're plotting the allele that's got the deviation from the norm, right? In particular,
yes,
I mean, it's kind of arbitrary, which whether you're going away from a norm. But if you're doing
right.
the allele that's actually showing the effect, then you would just call it an associated allele frequency.
That's a good
right.
Thank you, Ryan.
terminology.
Okay.
That's helpful.
Yeah.
Yeah, thank you.
We should ask you 2 weeks ago. Oh my God. Yeah, they really went off well. All right, I'm impressed. Good job. Any other questions for him?
It's late, so people.
Yeah.
Yeah, I especially liked your kind of summary at the end where you started to look at the bigger impacts potentially. I think that's really good. And do you have any, how hard is it going to be to cut? This is a loaded question because it's your thesis writing on the line.
Right.
How hard do you think it's going to be to code up?
Well, it depends. Some parts of this I've been doing already on an ad hoc basis because Jerry will make these information requests. He said to me the other day, Ari, I want you to look up the levels of predicted chromatin functional state from chromimpute in 6 cell types in 232 SNPs. And he wanted me to do this manually in the genome browser, and instead I
No, I didn't want you to. I said.
No, we
can do it however you want.
We just needed it that afternoon. That was the problem.
I said.
So instead I downloaded some bed files and wrote a script. But if you, it's okay. I don't mind.
Well, this is really something. It's obviously you guys we're facing it together in bioinformatics.
Yeah.
I mean, we're getting flooded with this data.
There's too much data.
And
It's spurious.
in our field, so the one thing I guess didn't say at the very end, so I'll say it now, is that, the thing that's really made these GWAS studies go is they were done with well-defined patient populations and stratified populations. And that is absolutely key. So what we've got to do now is go out and validate these assignments by recruiting patients who have these diseases or conditions. That's an expensive process, time-consuming. We do have a network set up to do that across about a dozen sites in the National Network of Depression Centers. But it's really quite expensive to do it. We've got a randomized control trial on our Generation 1 test right now going. It's got an N of 364. The the randomized control trial is costing us $6 million. I mean, it's very expensive.
Yep.
So looking for ways to recruit the patient, because ultimately we have to validate these assignments in human populations, and that is because in order to get this out and be used, there's 3 steps. Step A, Ari and Jerry and myself and others in my lab are doing. It's related to the basic science and discovery. Step B is the clinical validation, which could have animal models, but rats and mice don't actually do as much good with this kind of stuff, especially for reasons that we can describe. It's human research, meaning we have to identify the patients, recruit them into the studies, phenotype them. How bad is your depression? What drugs have you been on? All this kind of stuff. It's really expensive. And then the third thing, to get it into the marketplace, is how effective is this? How effective would this be based compared to what's out there already? And what they want to do is see relatively large trials and see whether you're saving any money. I mean, none of this gets out there unless you're actually saving money and can demonstrate that.
So what is this that you want to get out there?
Well, what we're after is a new pharmacogenomic, combinatoric pharmacogenomic assay for psychiatric disease. We work closely with a company that has owns that market space right now with exomic, variants across about 12 genes. But we're seeing that most of the interesting stuff is not happening in the exome. So and that was my point about those Stephen Johnson SNPs, and I really wanted to ask Jerry about that because, are the SNPs that are being tested for in Singapore and now in Taiwan, are they the intragenic SNPs that we found?
Yeah,
or
they're the same exact
They're
SNPs.
the same exact.
So if I could just close, just comment briefly, there was a question about how difficult this is going to be to write.
Yeah.
A lot of this data comes in the form of bed files, and of course, juggling GWAS is a matter of existing.
Yeah, on the line.
Those parts are easy. Some of the neuroanatomy parts we're just going to have to plain toss out of the automated portion because you can't do them on an automated basis. Perhaps the hardest thing that I'm going to automate is the high C portion, where I'm actually writing custom asymmetric binning high C compilers to compile on the fly to find the interactions for specific regions that are not captured by existing high C bins. So there's greater and lesser difficulty, but one of the things that is going to end up happening is tossing certain parts of the analysis out because they can't be automated. Because the goal here is to produce a tool rather than a philosophy.
Go.
So thank you very much, and I think we're going to close.
Thank you, Ari.
Known drugs and known targets.
That's right.
I think